First of all, I would also like to thank the organizing committee and especially Eva, who invited me here to give my presentation. And again, my presentation will be like a step back more to the basic science, because I am studying just the protein and some factors that can impact its stability, as the title of my presentation says. So, brief introduction, sorry for that. We all know that proteins, they are extremely important because they play so many roles in every living cell. So, and because of that, we all would like them to be stable, to be functional, and just behave. But unfortunately, in the case of many of them, they are like this mimosa or touch me not plant. When treated inappropriately, they will fold, unfold, they will change, and then we can experience some adverse effect. And there is so many factors that can have an impact on those proteins. They are of both extrinsic and what is the laser here? Nature. So we have to look carefully at the solvent and temperature, the radiation, intramolecular interactions, the pressure, including the cellular osmotic pressure. But there are also some factors that are could be kind of encoded in the protein itself. This could be some mutations which may happen spontaneously, or they could be already in the genetic code. There are also some specific sequences you can find in many proteins that can be involved in the aggregation. Also some secondary and tertiary structure connections, like local conformational strains or disruptions in the intramolecular interactions. So when any of those factors happens on the way of the protein, we can move from the known and very expected fragment of the protein folding channel leading to the native state towards this unwanted one, which is the amyloidogenic one. So once we enter that part of the folding channel, we can eventually find ourselves somewhere here on one of those aggregations roads, leading finally to the amyloid fibril or different kind of oligomers that can be more or less toxic. When we happen to be here, then we are in trouble, because one of those situations may happen and we can develop one of those neurodegenerative diseases we are all interested, all participants of the symposium are interested in. The protein I'm studying is not on that slide, and it's not on that list, because I'm interested in human cystatin C. It may be not the first amyloidogenic protein which comes to mind when you are thinking about amyloid and neurodegeneration. However, this nice small 120 amino acid protein is nice enough to behave when you treat it properly, but also you can quite easily push it to the amyloidogenic way. So by that, it is also involved in some neurodegeneration. For once itself, it can. It will come on the next slide. It aggregates, but also it plays some role in the development of Alzheimer's disease. Because it was shown to interact quite well with amyloid beta peptide and also the APP protein, preventing for some extent the aggregation and the release of the toxic amyloid beta peptide. But as I said, it also linked to neurodegeneration because the wild type protein is a component of some deposits found in the central nervous system, mostly in brain vasculature, and the point mutant, which is single mutant. This is single nucleotide mutation. It's responsible for amyloid angiopathy, observed mostly in the Icelandic population, but sometimes also spreading. This is the inherited disease, so it's almost extinct. But the exact mechanism of the cystatin C oligomerization is not known yet, but there are some hypotheses. First of all, as Eva already mentioned at the introduction, the protein has very nice fold, which consists of this beta strand and the perpendicular alpha helix, and similarly to Eva's stephin B, the protein can open and then it can enter one of those oligomerization routes, leading finally to fibrous or bigger oligomers. It can be by association of dimers, but also it could be this runaway mechanism. So we ask the question which one of them could be the case, but also what are the factors that can push this nice protein into the oligomerization. We look at the protein, and there are some intramolecular strains. First of all, there is a loop one, which is called the spring loop, because or molecular spring, which has the valine in position 57, which is a little bit off in the stability and the conformational angles values. So it forces the loop to be a little bit constrained, and then no wonder that it would like to open the molecule and then connect with another ones. And the second place is the 68 position I already mentioned. So the native leucine, which is green here, maybe it's not well visible, is substituted by glutamine, which is bigger. And then it expels the alpha helix from the core of the molecule, and we have the propagation of amyloid formation. So we decided to rescue the protein. So we did some point mutation. We have exchanged the valine 56 into other amino acids, which are more expected to be in the loop structure like this. And we have also tried to look at the 68th position. However, out of the 57 mutants, we succeeded to obtain all designs, but the 68th position, only one of the mutations turned out to be stable enough to be studied. So as you can see, the design mutation, which were thought to stabilize the protein, and we proved our concept by obtaining a couple of variants which were stable enough, which were more stable than the wild type protein. During incubation, the wild type protein dimerizes, but our variants not so much, I would say, except for one which is so stable that it doesn't really want to move or change its structure even at such a harsh conditions. The 68 variant, it behaves something in between, like a wild type protein and between the amyloidogenic variant. So we also checked the oligomerization, because dimerization can be the dead end of the fibrillization. We have also checked the oligomerization of those proteins, and we could see that in contrast to the wild type protein, I hope you can see something here. Those are short protofibrils. They are short and quite thick, so they have to get transformed into major fibrils. But in the case of all the other mutants, we don't see such effects. So the oligomerization was also lowered, or the propensity to oligomerization was much lower for all those variants comparing to the wild type protein. Our proteins were stable enough to get the crystal structures of them Before our attempt to the scientific words, only the dimeric structure of cystatin C was known, and we were lucky enough to crystallize those proteins in the monomeric form, which gave us much more information about the shape of the molecules and basically confirmed the shape which was suspected before. And we can also get the structure in solution, which is even more valuable because it gives you the dynamic situation of the protein. So you can draw some hypothesis what is happening when the protein, how it behaves in the native environment, which is aqueous environment. This is the picture showing the interactions with the papain, which is one of cysteine proteases. It's a plant protease. I also mentioned that some specific sequences can be already encoded in amyloidogenic protein. There is one of such motifs which is called steric zipper. It was introduced by Eisenberg, and it is a very small, short stretch of amino acids, up to 6, 7. It's exposed on near the surface of the protein, and it forms very close and very strong intramolecular connections using the side chains of amino acids, and those structures can be like a seed for further propagation of the fibril formation. There is a method to predict steric zipper presence in the any protein. To do this, you just have to submit your sequence to the group. It's free. It's for free. They do it for free, and they do the calculations, and they provide you with graph like that, histogram like that, which shows the red regions of the protein, which are supposed to be those aggregation inducers. In the case of cystatin C, we have two regions. The first one, which is centered no longer around the 57th position, and there is also the C terminus. We have focused on this, on the red one, which is here in the structure. We have obtained a bunch of peptides consisting of 6 amino acids, covering basically the whole sequence. And then we subjected them for fibrillization studies, and we were able to show that indeed such as short sequences are able to form regular structures. They resemble more or less either fibrils or microcrystals, and the fibrillization propensity is strongly sequence dependent. So some of them are stronger and some of them are weaker inducers of the fibrillogenesis. As you can see here, depending on the structure, we were also doesn't work? It does. We were also able to do some fiber diffractions on this material I showed you on my slide before. And by doing this, we were able to confirm that this beta structure, which constitutes the core of every amyloid fibril, is present in those short peptides. This is the specific pattern on the this is the specific diffraction pattern, which shows us that there is indeed this cross beta spine present, which could be the driving force for cystatin C self-association and fibril formation. Because a molecule, it's not isolated, so we also should check some environmental factors that can be involved in its aggregation. One of them is our membranes, because proteins, they have to cross membranes when they are secreted, and cystatin C in general is extracellular protein, and during its trafficking it crosses a couple of membranes. And membranes were proposed to be also involved, responsible from amyloid fibril formation and also for neurodegeneration. Due to different mechanisms. So we decided to check if mimetics of plasma membranes can be also involved in fibrillization of cystatin C. To do this, we used mice cells, which were composed of two different lipids because they mimic better eukaryotic membrane. And we were surprised to see that our mice cells, they don't induce aggregation, but on the contrary, they were able to shift the equilibrium for our proteins from the dimeric side towards the monomeric side. So it will kind of inverse the situation. We have checked this for this dimeric cystatin C mutant, but also we have produced the dimer, which we studied, and we have also shown that we also can see the formation of monomer here. And at the same time, the formation of monomer was associated with small changes in the secondary structure of the protein. We could see the increase of the alpha helical content, and as Eva already said, the fibril formation is associated rather with beta sheet formation. In our case, it was the opposite. So it shows us that indeed there is some unexpected effect. We were also able to model and to establish the binding mode between cystatin C and the membrane mimetics. So we could see, thanks to the animal experiments, that lipid binds close to this loop, which is critical for protein stability. And the final factor, we were studying are metal ions. So metal ions, especially the redox active metal ions like copper, they are accused of being one of the factors that can be responsible for neurodegeneration and protein aggregation. And to interact with a molecule, there should be some residues in the molecule that can catch this metal. In the case of most protein and copper ion, these are histidine residues, and in the sequence of cystatin C, we have 3 of them. 2 of which are located quite nicely in this region of the protein, which is not so compactly folded, it's more flexible. So we decided to check if cystatin can bind copper, and it did. It does bind. It can bind 1 or 2 copper ions, and the binding of the metal ion, it doesn't cause much changes in the molecule. However, it changes its stability. It changes the stability, and it causes the protein to precipitate quite quickly from the solution. So we decided also to check which of those 2 histidines is more, if one of them can be more strongly involved in copper binding, because as you could see, there were 2 copper binding places. And also, again, we did some protein expression and we obtained the variants. And as you can see, again, no impact on the protein structure. However, there is an impact on protein stability, and impact suggests that the histidine in position 86 is much more involved in the copper binding because when we removed that protein, that residue from the protein sequence, the protein was much more stable than when it was still there. So we also checked the propensity for dimerization and oligomerization and fibril formation for all those proteins. So we could see some decrease in fibril formation, fibrillization, as the thioflavin T assay shows us. And again, it was all consistent with the major role of the histidine 68 in copper binding. Also, the experiments, the animal experiments, especially again, NMR experiments allowed us to map the copper binding site here in this region. But more interestingly, we could use the titration with such a small system, which is tripeptide coordinating with copper ion, to single out one of those histidines and confirm that indeed the histidine 68 can create the ternary complex with this small system. Therefore, in solution, we could see that indeed the histidine 68 can be involved, and by these interactions can be some intramolecular interactions stimulated. We also did the mapping of copper binding site using crystallography. We couldn't obtain the crystal in the presence of copper ions, but we did some detour, so we obtained crystal of the cystatin C, and then we soaked the crystal into this in the solution with copper. What we could see after some time, there was additional electron density in the presence of one of the histidine residues, but when we looked closer, it turned out that this is the other histidine, so we were surprised. Anyway, when we looked at the neighborhood of those histidines, we could see that histidine 86 is not available for copper binding because it interacts with other residues, but it also shielded by PEG residues. So in crystal, the situation is maybe not that relevant. So to sum up, by our studies, we could have a closer look at the molecule and study some both external and internal factors that can be responsible for its stability, and then we can add all those information we got from our studies to draw some conclusions how to deal with amyloidogenic protein, what we have to look at, and also try to design some inhibitors of the processes. So the basic studies are very important if we would like to understand what is really going on into pathological state like neurodegeneration. So thank you for your attention.